Picking up right where we left off with the structure of the DNA double helix, we were talking about how small-scale structural details of the functional groups that compose the molecule, such as the fact that phosphates are negatively charged, give rise to these macro-scale important structures, the idea that DNA is overall a highly negatively charged molecule. We are also pointing out that the DNA double helix is composed of a minor groove, see this narrow channel, and a major groove on opposite sides of the DNA molecule, and on the DNA double helix. The important idea here simply being that this is an example of how small-scale structural details give rise to big structural features. Here now we're talking about the major and minor groove emerging from the presence of the kind of pucker that the ribose moiety in the DNA backbone adopts. Let's look closely here. There you'll see the ribose, all the way around through the molecule. If we back up, you'll see that here I've drawn the ribose, or deoxyribose to be more correct, as a planar molecule. And of course you know that since it's composed mostly of a bunch of single carbon bonds, it's not planar. In fact, it has to pucker. That pucker can either be at the C2 position, right, carbon 1, carbon 2, carbon 3, carbon 4, or at the C3 position, so that's C2 endo or C3 endo. C2 endo gives rise to this B-form DNA, which we're looking at here. That's the heavily hydrated form of DNA that most commonly is present inside the cell. C3 is a less common configuration, A-form DNA, and I'll refer you back to your textbook if you want to see what A-form DNA looks like. But again, all of that simply arising from the configuration that ribose molecule takes on. Another interesting feature of the double helix, of course, is this intra-strand packing. So here's one nucleotide, and here's one nucleotide away. There's forming some nice hydrophobic as well as some interesting polar interactions there between bases. Let's turn briefly to RNA before moving back to proteins. Here again, a nucleotide polymer, except that in this case it's actually a ribose, not a deoxyribose. So we haven't lost this hydroxyl group that is the telltale of deoxyribonucleic acid. Instead, we have a ribose, ribose phosphate connected by these phosphodiester bonds. So very similar structure to DNA, with the critical difference being the presence of this ribose, the presence of this hydroxyl, which can actually act as a nucleophile, performing a nucleophilic attack on that phosphate, and rendering RNA to be a much less stable molecule than deoxyribose. However, without going into too great detail on the origins of the RNA world and life in general, let's just focus on the idea that, like DNA, this unique sequence and structural information of RNA emerges from the sequence of these nucleotide bases. All right. Now, we've already said that RNA plays a more complicated role than the threefold role that I'm suggesting here, but this is a sort of a standard textbook way to think about RNA. Messenger RNA, of course, being the transcript of the DNA read by polymerase from the genome itself. Transfer RNA reading that sequence of the messenger here, right, codons matching up here, and the transfer RNA being charged with amino acid that can be added to a growing polypeptide chain on the ribosome. So there's transcription of DNA to RNA, there's transfer of that information by a tRNA to the ribosome. Here's the ribosome, part of the ribosome itself. And as an aside, I'll just remind you that the ribosome is this complicated nucleotide. Here, gold is the RNA of the ribosome, and in blue are the proteins associated with that nucleotide polymer, nucleic acid polymer, excuse me. And then here's the transfer RNA. Again, that matching up of codons with the message in red here happening here, and then the extension of the protein polymer happening here. So the ribosomal RNA plays that third role in catalyzing the synthesis of the protein that goes on to do the work of the organism. Now, we've alluded to the fact that there might even be more roles for RNA, and of course we expect that the story is much more complicated than this, but we won't go into that today. Instead, I want to turn quickly to proteins before we move on to our next subject. Proteins are going to be the focus of most of our study in this course, and we start off with this idea that a lot of interesting biological molecules happen to be polymers. Let's start with the DNA, though. Proteins are encoded at the DNA level by three-letter words composed of the nucleotides of DNA, A, T, G in this case, encoding now a methionine. So what we can do is go from a simple 4-letter code in DNA to a 20-letter code for proteins, which of course referring to the fact that there are more or less 20 naturally occurring amino acids that we find in proteins. Sometimes there are more, sometimes there are fewer, depending on what kind of system we're working with. But generally, we'll be studying 20 amino acid side chains. So ATG encodes for methionine, right, represented in the single letter code M here. CTG represents a leucine. CCC represents a proline. So we can see how we can go from this 3-letter word to a sentence that defines our protein polymer. We'd write the protein sequence this way, MLP for methionine, leucine, proline, but when we're drawing the chemical structure, we see some interesting details emerging that remind us of what we've already seen for other polymers. So here we see amino acid 1, that R group that defines its identity as the methionine, we're just ignoring for the time being, instead noticing that amino acid 1 is linked to amino acid 2 via a peptide bond, right, linking a carboxylic acid to an amino group through this peptide bond or amide bond, right? Again, that's one of the functional groups that I'm expecting you to recognize. And then we see amino acid 2 linked to amino acid 3 by, again, a peptide bond. So we're going to talk in detail about amino acid structures, but just to give you the idea that there's a backbone, a repeating backbone, just as we saw for the nucleic acid polymers, although now it's composed of amino acids bound together through peptide bonds. Again, saving the details of structural information of amino acids and proteins for another lecture, we'll simply wrap up by saying this sequence now of amino acids, right, not nucleotides, but sequence of amino acids determines the structure and function of the protein. So here's the sequence of a protein known as green fluorescent protein. Now, this might be one of the first examples I've given you of a so-called ribbon diagram of a protein. I don't want you to be threatened or confused or frightened by that in any way. We're going to define these ourselves in subsequent lectures. For now, let's just take it for granted that this is a common representation of a protein. The protein chain starts here and winds all the way around, and what we're doing now is just following the backbone structure of the protein, ignoring all the amino acid side chain information. However, it is the details of that sequence, all of that side chain information, that drives us to form the structure of green fluorescent protein. This is another protein called crustacyanin. It's another pigment molecule, also from a sea creature, although now from lobster and other crustaceans. And this amino acid sequence defines that protein structure. The key here is that one sequence determines one structure. We don't cross over, right? GFP is not going to fold into crustacyanin. Now, what I want to be careful to add here, let me hasten to add that there are always rules that we can break. And sometimes amino acid sequences will encode for multiple structures. We're going to talk about that later in the semester, but for now, the big idea is simply that the structure of the protein is encoded at the amino acid sequence level. That brings us to the end of this 4th video. Next up, forces. See you there.